Thanks so much, Alex, for organizing, for having me. I'm looking forward to the main talk by Jackson Loper, who's visiting us from New York. And I'm just going to get us started by giving a little primer on something that I'm sure many of you are familiar with if you work in computational biology or data analysis, or even if you've just taken a stats class. Some of this may be very familiar, but it's so much kind of in the background underpinning the ideas that Jackson will talk about. It's been a while since we had a primer. Manny Rivas, now faculty at Stanford, gave one on multiple testing a while back, but these are meant to be pedagogical, and maybe we haven't had one in a while, and maybe this will be a little bit of a different perspective. So here we go, a primer on multiple testing. But before we talk about multiple testing, I think we should just talk about testing one hypothesis. And if this feels like Stats 101, it's because it is. That's fine. But, you know, it's morning. We can kind of wake up and start with things that hopefully we're somewhat familiar with. Null hypothesis significance testing, I'm not even going to write it out, is the framework that's, you know, although it's been widely criticized and there's all kinds of other thoughts and feelings about this and p-values and Bayesian decision, and nevertheless, this remains the standard in science. And it was introduced by Fisher back in 1925. And the current framework is also due to work by Neyman and Pearson shortly thereafter. And we consider a null and an alternative hypothesis. And I'm going to do everything I write on the board. I'm going to kind of, if I can take a simplifying assumption so that it's easier to present, I'll do that. Okay, you can always look at a textbook for sort of more general formulations. And so for the first example of that is that I'm just going to take a point null, where the null hypothesis is that some parameter is equal to some null value, theta equals theta naught. There, you could do a more complex null where it's in some null region, but that would make certain things a little harder to write. I would have to take maybe some supremum over that region in certain cases and things like that. But if you want to see a very nice kind of elementary reference, one that doesn't have all the proofs, but a great place to start looking, if this, especially if this is not as familiar to you and you'd like to get up to speed, is Larry Wasserman's All of Statistics. It's a tongue-in-cheek title. It's a good book. And so the alternative hypothesis would just be the complement theta is not equal to theta naught. And in many settings, we think of this one as no effect, and this one is, yes, there's an effect. Okay, there's some difference between the case and control groups. You know, these genes are differentially expressed. This allele is more frequent in cases than controls. Some examples from biology. But, okay, and then how would we go about testing that? Well, we have a test statistic, which is derived from our data. Function of data. And we have some rejection threshold. Again, you could be more general. You could say there would be some rejection region, but I'm just going to take a simple and very common setting where there's some value C. And how low can I write here? Is this blocked for anyone? It's okay? Okay. We reject the null hypothesis when the test statistic exceeds the threshold. Very simple. And what do we say otherwise? Otherwise, the way we talk about it is that we retain the null. So we reject the null or we retain the null. And so it's sort of like innocent until proven guilty. We need a lot of evidence to reject the null hypothesis, and otherwise we accept it. And to emphasize that, we talk about the two different kinds of errors one can make, and I will make a little table for that. So what are the different scenarios? We could be under the truth could be the null hypothesis, or the truth could be the alternative. And given our data, we could retain the null or we could reject the null. And in this case, we correctly retain. In this case, we correctly reject. This is the most important kind of error in the traditional framework. It's the error of a false rejection, also called a type 1 error. That there's no effect, but we incorrectly reject the null and therefore think that there is evidence for an effect. And the other type of error is a sort of false negative that we miss. It's also called a miss. Okay, there's a true effect, but we missed it. Okay, and then we can talk about the power as a function. Sometimes denoted beta, of the true parameter theta, and that is that the probability under that parameter that we reject. Okay, so the probability of rejecting depends on the true effect. For a stronger effect, we're more likely to reject. That's the intuition. Okay? The significance level of the test is the value of the power function at the null hypothesis, at the null value of the parameter. Okay, so this is the probability under the null that we, in this case, falsely reject the null hypothesis. So this is the probability of type 1 error or false rejection. Okay, and this is a property of the probability model and also a property of the test statistic and the rejection threshold. Okay, and please interrupt if you have any questions. And the classic choice is, of course, famous, infamous 0.05 for the significance level, right? Which means that the probability of a type 1 error is 1 in 20. And Fisher did introduce this, but interestingly, Fisher also said this was never meant to be a fixed choice. That he advocated back, I think it's a paper in the 50s that this should have always been context dependent, and well, people like simplicity. So it's become a kind of. I'm not going to make more comments on this. There's a lot of writing on it. You can find it. It'll take us too far afield. There's more to talk about. Other things to talk about. So generally, you might have a family of these tests, and you can consider lots of different values of the threshold. Okay, so a family of thresholds, C alpha, where alpha is between 0 and 1. And I'm labeling each one by its significance level. Okay, so alpha is the significance level of the test with threshold C alpha. And the picture here is, if this is the distribution of the test statistic under the null, then this, if this is C alpha, then this probability is alpha. And finally, I'm going to introduce the p-value. The p-value for an observed test statistic. Now, I've been writing the test statistic as a random variable. Now, just suppose that we have some actual value t. Or let me write t equals t. Okay. There's different ways to write this, but one is the infimum, the lowest possible alpha, such that you still reject. Okay? Lowest level, significance level, at which we reject. And another way to think of it is this, as the strength of evidence. It's not the effect size. We didn't talk about effects at all, but I mean effect sizes. This is generally a property of both the effect size and the uncertainty that one has in the effect size. But another informal but ubiquitous phrase one hears is strength of evidence. Strength of evidence against. And one more formulation of this that is very useful is that P is actually the probability under the null that the test statistic would be greater than the observed one. So it's also the probability under the null of observing test statistic at least as extreme. And one more little note about p-values. What is the distribution of the p-value under the null hypothesis? If you might know this by heart. It's a good thing to know by heart. I would even say maybe you should know it by heart, but I will derive it or remind you. So for some alpha, the probability under the null that p is less than alpha, if you look at this definition, it's equivalent to the test statistic being larger than C alpha, which, by the way we define C alpha, is alpha. Okay, so the probability under the null that the p-value is at most alpha is alpha. For alpha between 0 and 1, what do we call that distribution? The uniform distribution. Good. So conditional on the null, p-value is uniformly distributed between 0 and 1. And a little asterisk here is that that's true for continuous test statistics. For continuously distributed. Okay, if you have a discrete test statistic, it's not quite true. You have to be a little careful because you actually can't realize all possible C alphas exactly. You have some subset of the possible values, and this will be some discrete approximation of uniform distribution. So if you have like a Poisson count or something, then yes.
Oh, that's great. Does it only depend on the test statistic being continuous versus discrete, or does it have anything to do with how much data we have? Like, what if we're in some pathological case where we have 1 data point or 3 data points? Can we still achieve any possible p-value?
Good question.
I think we can. Yeah,
I think that is correct. So I think what I wrote is still correct if we talk about the distribution of t given the sampling scheme. So the number of data points you collect is part of the probability model here. And then, yes, I definitely agree. Okay, so let's move on to the promised topic of the primer. And the first I'm going to talk about 2 different ways to think about multiple testing and ways to control it. The first one is the most straightforward. Okay, I will tell you what that is in a second. But we're testing not one hypothesis, but multiple hypotheses. Okay, we have a whole bunch of hypotheses, and it's sort of almost semantic how you think of this. You could think of it as many hypotheses on the same data set, or maybe you have one hypothesis that you're testing on many repetitions of an experiment, or slightly different data sets. It doesn't matter. I have a bunch of hypotheses I'm going to test. Again, I'm going to index them with a little superscript. So H0I versus H1I. This is the ith null and the ith alternative. And I'm going to have m of these. So I will go from 1 to m. And the simplest thing the simplest and most stringent thing that we could try to control is the probability of making even one false rejection. That even one of our tests had a type 1 error. And that's called the family-wise error rate. The family-wise error rate, FWER, is the probability that we make at least one false rejection. Okay, and this is the most stringent control. In the multiple testing setting. And there's a very simple method that always works. People are nodding. People have heard of this. It's the simplest method. It's called the Bonferroni Method. And one takes the p-values corresponding to the tests. And you reject the ith null hypothesis if pi is less than not alpha, but alpha divided by m. Okay, you adjust the significance threshold by dividing it by the number of tests. And why does this work? It turns out this always works. You do not need independence or any other assumption. And the reason that it always works is because the proof involves something that is always true, which is called the union bound. Okay, and I'm going to show this to you. This is the only thing I'm going to prove in this. Well, I guess I kind of proved this uniform distribution thing. But this is the only thing I'm really going to prove is this Bonferroni, because it's a one-line proof. So what do I mean by it always works? I mean that the family-wise error rate is at most alpha if you do this. Okay, so let me prove it for you. So let AI be the event that the ith hypothesis is falsely rejected. Okay, then what is the family-wise error rate? It's the probability that any of them are true. So that's the probability of a union, right? But the probability of the unions of some events is at most the sum of the probabilities. Worst case scenario, they don't overlap at all. And in that case, what is the probability of one of these false rejections? It's alpha. Well, actually, no. It's alpha over m. Yes? Okay, so this is m times alpha over m, which is alpha. Okay, that's the proof. You don't need independence. If anybody tells you you need, well, you can do it if you don't have independence. If the events or the p-values are kind of correlated, if they're positively correlated, this becomes overly conservative. That's the intuition, and why we might seek alternatives. I do want to say one thing about the independent case because it's so important. It's pretty good in the independent case. It's not perfect, but it's pretty good. And for that, I want to look at 1 minus the family-wise error rate, which is the probability of no false rejections, which is an intersection. This is called De Morgan's laws or something. Probability that none of these events hold. And that is the probability of an intersection under independence is the product of the probabilities. So here we get 1 minus alpha over m to the m, which is approximately 1 minus alpha for alpha small. Okay, there are additional terms, but which basically says that in the independent case, if these events don't have too big of a probability, then the overlaps are very small, right? They're quadratic. If this has size alpha, the overlaps are roughly alpha squared, and that's kind of the next order correction is on the order alpha squared. So in that case, you don't lose too much in the union bound. And I'll just write, for the record, positive correlation. I'm not going to make that precise. Between the p-values, it'll be overly conservative. And I mean, the extreme case is if you're testing, if a whole bunch of your tests are actually testing the same thing exactly, then a whole bunch of your p-values will be exactly the same. Then you shouldn't have divided by m. That's silly, right? So that's the extreme case is that you're just doing repeated testing of the same thing. And something somewhere in between is sort of a key example of all this, especially in this building, is genome-wide association studies. I'm going to say a few words about this and how it relates to family-wise error rate and Bonferroni correction. Oh, good, we're fine. Okay, so how many of you have seen a Manhattan plot? I actually would like to see a show of hands. Okay, so if you haven't, now's the time. They're all over the internet. You can Google it on your phone while I'm talking. But I'll just draw a sketch of one so we have it in front of us. This is where you plot. Okay, so a genome-wide association study, you are looking for associations between genetic variants and some phenotype. So does having the alternate allele at this site increase your risk of some disease or increase your average height by some tiny amount or something like that? And what we do is we just do these simple marginal regressions of the phenotype on the genotype. We don't do some crazy joint model. It's the simplest thing you could imagine. You just look at one genotype at a time and you say basically in the case control setting for a disease, is the allele frequency, the frequency of the alternate allele, say some people have an A and other people have a T, is the frequency of the A allele higher in cases than controls. We just do some regression, and we plot the p-value for that regression. Actually, we usually plot minus log p. And here's the genomic location on the x-axis. And you get a whole bunch of dots. Most of them are pretty low. And then you have sort of fewer that are higher and even fewer that are higher. But but there's a pattern that I haven't drawn yet, which is that when there's higher dots, they kind of take some friends with them. And this is why you get these skyscrapers, and this is why it's called a Manhattan plot. And if you zoom into one of these, you see a little more structure. You kind of can see some basically spreading of the signal. And why is this? This is because genetic variants that are nearby on the genome are correlated. It's called linkage disequilibrium. It's a result of the way human genetic material is passed on from one generation to the next, recombination. And so you get these local correlations along each chromosome. And that means that if you have some causal variant over here, but this is the one you're looking at or measured, you didn't even have to measure the causal variant, that because they'll more often occur on the same haplotype, there will be an association with the variant you're looking at with the phenotype, even if it's not the causal one. And this is how you get these correlations that drive this Manhattan plot picture. And so, folks have been well aware of this. And there's this question of what should we do to correct for the multiple testing burden? Clearly, we'd be shooting ourselves in the foot if we use the naive Bonferroni correction for all the possible genetic variants, because there's so much of this local correlation. But Bonferroni is still sort of the right idea because if you go wider than this local correlation region, you do kind of have independence. Especially on different chromosomes, but even if you're far enough apart on the same chromosome. So it's kind of like there should be some effective M, some effective testing burden. And then the question was, what should that be? And this was worked out in the 2000s, although the ideas go back to linkage mapping. So I'm going to write the references down because they're really interesting to look at. All of these are great papers. Lander and Krugliak, Nature Genetics, 95. The ideas go back to this paper, which talked about a genome-wide significance level for linkage mapping. And then when people turned to the idea, when it started to become tractable to think about genome-wide association studies because we had enough data, enough genotype, started to become possible to genotype a large number of individuals, people thought about this question for GWAS, and the two key papers came out in the same issue of, I think, genetic epidemiology in 2008. One was by FWER and Mark Daly and a couple other authors, and the other was Frank Dudbridge. This is Itzik pair. And Gosnanto. And basically the idea was, let's do this empirically. So this is often called permutation testing, although it was not so much rearrangement as just kind of randomizing cases and controls. So I like to think of this as an empirical null distribution. And what did they do? They took a whole bunch of real genotypes. I'm not going to write this all down. And they had a few thousand genotyped individuals, and they randomly assigned them to be cases and controls. So there's no true signal. The phenotype is a null phenotype. It's a random phenotype. And then they did a genome-wide association study with this random null phenotype, and they looked at what was the lowest p-value, the most significant, the highest on this minus log 10 scale. Then what is the most significant or the lowest p-value that you get in each of these runs of the simulation? And then you build that into a distribution. You do this many, many times. Actually, they did it about like 10 million times. And then you get this null distribution. And then you can ask what it looks like. So random phenotype. And then look at distribution of the lowest p-value. And actually, there's one more subtlety, which is that they didn't yet have a dense set of markers. So they wanted to extrapolate to what it would look like if they were really looking at every possible site in the genome, maybe at a given allele frequency threshold. So what they did is they downsampled the markers to see what the curve looked like. And you had fewer to extrapolate to what it would look like. Turned out there was a kind of asymptote for the effective testing burden because of this correlation. If you only look at a few variants, they're kind of independent, so the effective testing burden, the effective number of independent tests will grow linearly at first, and then eventually it'll asymptote. It's okay if that was a little technical. I just wanted to give that little nuance for those who maybe have thought about this kind of thing a little more. And what did they find? They found a genome-wide testing burden on the order of 1 million. And that means that the genome-wide significance threshold, if you want a family-wise error rate of 0.05, should be 5 times 10 to the minus 8. And this became a community standard. Now, there are some caveats, which is that this depends on the demography. If you do this in African individuals, you actually get a number about twice that if you compare it to doing it in European or East Asian individuals. And that's because genetic diversity is higher there. Because of the bottleneck, when the out of Africa bottleneck, the LD is much longer outside of Africa. The LD is shorter inside of Africa. There's much more independent genetic information along the genome. Another caveat is how low are you going in terms of the common variants you're looking at. You never look at super rare variants in GWAS, but you might look at common variants down to some allele frequency cutoff, maybe 5% or 1%. It should depend on that too. Nobody cares about all that anymore. They've decided, let's just use this. It's a community standard. It's a line in the sand. And it's been very successful in sort of ensuring that genetic findings are reproducible. So I think it's widely considered a successful community standard. That before this there were all kinds of non-reproducible results, and after this that after this became accepted in the community, that sort of ceased. Good. Now, the family-wise error rate is often a little bit too stringent. Oh yeah, I hate this. Is there a microfiber cloth? Yeah. Okay. The family-wise error rate can be a little bit too stringent in settings where we're willing to tolerate some type 1 errors, especially if we have sort of a lot of tests and we think a whole bunch of them are actually probably true. And maybe it's okay if occasionally we have some false ones mixed in with the true ones of the ones where we reject, right? You're going to help? Okay, thank you, Brian. Thank you very much. Okay, so actually that would be especially great if you continued to erase because I'll need a couple more boards. I'm going to start writing over here. Thank you. Okay, and so Benjamina and Hochberg introduced this idea in the mid-90s of the false discovery rate. And this is the mean fraction of rejections that are false. Okay? And so let me formalize that a little bit and let's look at this table of the things that can happen when you're testing multiple hypotheses. These are I'm gonna count all the different, I'm gonna write variables in the entries of this table that are the counts of how many times the thing happened. And I have the true null, the null is false, this is the alternative is true, column for the total here, total, and then this is the case where you reject. Sorry, let's start with retained, and then rejected. Okay, it's actually the same as the table you had before, but now I'm going to write the counts instead of the names of the things. And so I'll just use the notation that they used. M total, M naught are actually true nulls. This many are alternatives. Call that M1 if you want. And the false discovery proportion is defined to be the fraction of rejections that are false, because I rejected, but the null hypothesis was true, so that means it's going to be V over R. But there's always a chance that you don't reject anything, so you have to account for that. So I'm going to define it to be V over R when that's defined. So when there's at least one rejection. And 0 when there are no rejections. Okay, that's the false discovery proportion. This is a random variable. The false discovery rate is its expectation. And a couple quick notes about this quantity. One interesting one is the family-wise error rate is the probability. What is the family-wise error rate? That's the probability that there's at least one false rejection. So that's V is the number of false rejections, right? And so that is the probability that the false discovery proportion is positive. So they're not the same, but they're related, sort of. Family-wise error rate is the probability that this quantity is positive. The false discovery rate is its expectation. Okay, and this is a much weaker quantity to control in general, But there's one very interesting case where they're actually the same thing, which is under the global null. So under the global null, so that means all the null hypotheses are true. M naught equals M. In that case, V is equal to R. Okay? And the false discovery proportion is simply equal to 1 whenever there's at least one rejection or false rejection, and 0 otherwise. So it's the indicator function of this event. Okay, and in that case, the false discovery rate, what happens if I take expectations on both sides here, I get the probability that V is greater than 1 on the right, which is the family-wise error rate. Okay, so they're actually the same under the global null. And in general, intuitively this is probably obvious, but in general the proof is that the FWER, the family-wise error rate, is always more stringent, is that the false discovery proportion is always at most, since V is at most R, the false discovery proportion is in general less than or equal to this indicator function. So in general, FDR is less than FWER. And there is a method to control this quantity. There has been other work since, but introduced in the original paper was a very elegant and actually quite simple procedure. Sometimes it looks more complicated on paper than it is. This is the last thing I'll cover. I know we're running out of time. The Benjamina-Hochberg method. Okay, so what do you do? How do you control this? This is totally non-obvious. I'm not going to prove it. But it's straightforward enough to think about as an algorithm. And so what I do is I order the p-values for the tests first of all. I'm going to write the subscripts in parentheses to indicate that they've been ordered. These would not be the original indices. There's no reason they'd be ordered in their original indices. Okay, and what I'm going to do then is I'm going to do this funny thing where maybe I'll draw the picture first and then write the formalism over here. So I'm going to plot the ordered p-values. Okay, here is the index. Here's the p-value. I'm just going to draw them, and I'm going to compare them to this line. And the line is going to have slope alpha over m. So here, this here is alpha. This is alpha over m. And suppose that here's the first p-value. Maybe this is the second one, a little higher than maybe the third one jumped up a bit more. So the fourth one, fifth, sixth, so on. But suppose actually that this is the last time it was below the line. They might go above and below the line a few times, but what I look for is the last time that it's below the line. That's the rule. It's an algorithm. I'm not going to prove this. What you do is you look at the last time it's below the line, you call that the index I star. Okay. This value is the p-value at that index. Okay, so p paren subscript I star. And that's going to be the Benjamina-Hochberg threshold. Okay, so let me write it down over here now. So you let I star be the max, the largest index, the last time going forward, that ordered p-value is less than I over m times alpha. And then you simply reject the ordered null hypothesis I. I've reordered the hypotheses along with the p-values now, okay? I reject all of them up to that point, whether or not those are below or above the line. Okay, so, or another way of putting it, if you want to go back to the original indices, is that I reject whenever the p-value is less than this BH threshold. The threshold is data dependent, which is kind of funny. That's a property of this algorithm. And then what are the results? I said I wasn't going to prove anything. I actually haven't claimed anything. But now I'm going to claim things. So theorem, this is from the original paper. If the p-values are independent, and they won't be independent once you order them. Order statistics are not independent, they're ordered. But if the original p-values are independent, then using this procedure under the BH procedure, the false discovery rate is at most alpha, and it's actually controlled even slightly better by alpha times the fraction of true nulls. Okay, and then this is under independence. That's kind of strict. People use this much more generally. There's a good reason for that. There was a follow-up paper, Benjamina Ucatili, in which they showed two things. The first is that you can relax independence somewhat. And I'm not going to say exactly what the condition is. It's called a positive regression dependency condition. It's a little bit technical. But actually, for applications probably most relevant is that you can also make this just a little bit more conservative, and then it works in general, in the totally general dependent case. Okay, so in general dependent case, modify conservatively, replacing alpha. Sorry, my writing gets worse and worse as the talk progresses because I'm rushing more. But you replace alpha in the procedure. So where here, this alpha, you replace it with alpha over this funny little harmonic sum, I equals 1 to m, the sum of 1 over I. Okay? This is their, they have this cute trick that allows them to prove that if you do this, it works in general. So then still have the false discovery rate controlled by alpha. Okay, and I was going to talk about some further reading. I won't make those remarks now. I want people to get breakfast. Can post those on the website or talk to me after if you're interested in some other topics such as knockoffs, or Matthew Stevens. He has this paper called FDR, A New Deal. Besides being a great title, it's a great paper. Recommend that one as well. An empirical Bayes approach. We haven't really talked about Bayesian ideas here very much. This is very frequentist, old school stuff, but there are Bayesian perspectives on these things that are important and relevant now. But yeah, so this is, I guess the one other thing I'll say is that there's one other quantity, the last point, then breakfast. This is the false discovery rate. This is the family-wise error rate. A kind of compromise in a way, or a third quantity one could consider, is something that you could call the false discovery exceedance. Is that the right name? And that would be the probability that the false discovery proportion is greater than some value that's not 0, some q. Okay, and I think that might come up in Jackson's talk too, so I just wanted to mention it in the context of what we already had here on the board. Thank you so much. Let's go get coffee and breakfast.
So this is work I've been doing in collaboration with Wes Tansey and Li Hualei and Will Fithian. Li Hualei and Fithian are out at Berkeley. And as we go through the talk, I'll let you know what part they had to contribute. It was an amazing and cool part. Get excited. So this is work about multiple hypothesis testing, as Alex said. And I'm going to start with an example from cancer drug discovery. And we're going to think about this multiple testing problem. And we're going to see that the Bonferroni correction is a really bad idea for this particular problem. And then we're going to think about that, scratch our heads, and we're going to think about two different reasons why a Bonferroni correction to deal with the multiple hypothesis issues is the wrong choice. And specifically, we're going to realize that there's structure in the problem that we can take advantage of. And that's going to have two different forms. First, there's going to be a nestedness structure that we'd like to take advantage of, and we'll talk about that. And we can take advantage of that structure using techniques that are already known from the literature. And that's going to help, but it's not going to be as awesome as we hoped. We're going to cry, and we're going to think about what we could do to make it better, and then we're going to realize that there's something else. There's an additional kind of structure in the problem that we ought to be able to use, and that's that there's a certain kind of a smoothness that has to, in some sense, go along with the nesting. Because there's a nesting, there's also going to be a certain kind of smoothness, and then we're going to think, oh, we can take advantage of that smoothness also, and that's going to give us something that works pretty well for dealing with this problem. So then, once we're done with that, we're going to look at some extensions that don't involve this simple cancer case, but involve more complicated relationships between the hypotheses. This is going to lead to some discussion of an example with differential expression, which will be fun. And then finally, time permitting, we'll look at some plots, empirical results that show how the smoothing idea is actually, it's a good idea, and you don't have to take my word for it, but it actually does work. Or at least that's what these plots that I'm going to show you are going to suggest. So if you believe my plots, then you'll believe that the techniques that I'm going to propose today are effective. So this is sort of the story for what we're going to go through here. Looks good. So let's get started. Okay, so the problem, the example problem that we're going to discuss, the multiple hypothesis testing that we're going to think about, is you're thinking about different drugs that you might try to kill cancer cells. So there's a lot of different drugs. And there's also a lot of different dosages that you might try those drugs at. Okay? And you really want a low dosage if possible because you're giving these to humans. They're basically all poisons. You don't want to kill the human. You want to kill the cancer cell, but you don't want to kill the human. So you want to think about different kinds of drugs. You also really want to think about the dosages. And of course, you also want to think about what type of cancer you're treating. Okay? So that's a lot of different variables to think about, and there's a lot of different hypotheses that you have to test there. So, but there's these massive experiments now where they actually do it. They try all these different drugs, every possible combination of different drugs, different dosages, and different cell types. So they grab a little petri dish from their stash of immortalized cells that corresponds to some particular type of cancer. So these cells have been cultured and they have some resemblance to kidney cancer or something. They apply some particular drug at some particular dosage. They wait 5 minutes, and then they shine a light through the petri dish. And based on how much light gets filtered, that gives you some indication of what proportion of the cells died. Okay? So it's this kind of noisy indicator, but we're doing it at this massive scale, so we need something that's really going to work over and over again and be reliable. So this is one of the ways they do this. And it's a little bit of a noisy indicator of what proportion of the cells died, but it does tell you something about how effective the drug was at killing these particular cells. Okay? And then they do it with another cell type or a different drug or a different dosage, and they do that again and again and again and again. They get millions and millions of results. let's look at what some of them might look like, this data. So you're going to have different dosages. And then you're going to have this coefficient. It's a number having to do with how much light shines through, which has something to do with what proportion of the cells survived the treatment, survived the drug. And maybe you get something that looks kind of like this. At these dosages, there's a lot of noise on the line, technical artifacts, so it's not like you're going to get a smooth answers here, but you'll get all kinds of different things. Maybe you get something down here. Eventually, if you give a high enough dosage, basically all the cells die, and you can tell, because the survival coefficients are really low. So this is the data that we want to look at. And so to go back to the question of multiple hypothesis testing, the hypotheses we're thinking about is, at what dosage does this drug become effective? So this is a little bit of an oversimplification, but let's just say, for the moment, that for all these drugs, all these dosages, the drug has at least some effect. Okay? And for all these dosages, basically the drug has no effect. Now, of course, in reality, even the tiniest dosage of the drug is going to have some effect. So when you're really doing this, the hypothesis you want to test is that the drug has an effect which is lower than some threshold, and you have to make some corrections, blah, blah, blah. But to keep things sort of simple here, let's just say that the question we're thinking about is that the situation we're in is that for these dosages, the drug has some effect. For these dosages, the drug has no effect, and we're trying to find this change point. Okay, and to think about this, we do a bunch of control experiments at first at basically zero dosage. Maybe we just add water or something. And so we get a lot of samples at the zero dosage case. And then we can estimate some kind of distribution of what kind of survival coefficients we get if we give them no drug. Okay, so this is going to be the null distribution. And sort of more formally, the hypothesis we're testing then is that this observation, say if we're thinking about the hypothesis at this dosage, the hypothesis is that this observation comes from this null distribution. So in other words, the distribution of what kind of observations we're going to get at this dosage is exactly the same distribution that we would get if we had given no drug. So the drug is having no effect. That's the null hypothesis. And in this case, actually, there is an effect, so we'd like to reject that null hypothesis and say, yes, the drug does have an effect. Okay? So this process, a little more formally maybe, is we're trying to estimate this set S. All we get to observe is these observations, these controls, and we would like to estimate the set S of dosages at which there is some effect. Okay, that's our task. So the first thing we might try is something you may have heard of because we talked about it. It's the Bonferroni. So you may have heard of Bonferroni, but have any of you heard of Olive Jean Dunn? I ask you. So actually, fun fact, in terms of codifying this particular plan for estimating the set of dosages that have some effect, or in general, this kind of correction, this Bonferroni correction, it was codified in 1959 by Olive Dunn. So it's not really Bonferroni's correction. We've been lied to all these years. Alex and I were talking at dinner over this. We were just like, what is Bonferroni? What was he like? And we realized it's not even his correction.
It's his inequality.
It is his inequality. That's right. He has the inequality.
If they called it after her, that would be done.
That's true. Her name is Olive Dunn, and maybe if we named it after her, then that's a good point. Maybe it's best that we call it the Bonferroni inequality and the Bonferroni correction. So let's look at, in this particular case, what the Bonferroni estimator for S would be. So we're trying to estimate the set of dosages where there's some effect. We get these values. We have this control. What's the Bonferroni estimator? It's the set of dosages such that the p-value, which is the In this case, it's the cumulative distribution function of the null distribution applied to the observation. So this is, but you can just think of this as the p-value, individual p-value for each observation is less than, let's say, 5% divided by the number of dosages we're testing. In this case, low p-values are meaning that they are important. So in this case, it's F0. Yeah, I'm testing it from the left. So yeah, so if you're keeping very close attention, when Alex was talking, he was thinking about cases where the test statistic was really high, and that's when we reject. And in my case, I'm thinking about it the other way. In this case, if the test statistic is really low, we're going to reject. Right? You can test from either side, or you can test from both sides. But in this case, this is what the Bonferroni correction looks like. Okay, so basically what we're going to do is we're going to look at this null distribution. We're going to find the 5th percentile of it. Okay, say it's here. Say the 5th percentile is here, actually. Let me do it this way. Yeah, here we go. 5th percentile is here. Okay, this is not drawn to scale. So let's say this is the 5th percentile. And then we're going to divide the 5th percentile. We're going to look at, actually, instead of the 5th percentile, we're going to look at the 5th divided by mth percentile, which is a much stronger, smaller threshold. And we're going to pick all of the observations which fall below this Bonferroni threshold, the percentile of 5% over m. Okay, so here's what's Bonferroni is going to give us. S hat Bonferroni, or olive done. Okay, fine. Now, why would we want to use this? Well, as Alex mentioned, the nice thing about the Bonferroni estimator here is that we're guaranteed that it's going to control our type 1 error. So let's just, for completeness, say what that means in this case. For any given dosage that's in the Bonferroni estimator of the set of dosages where there's an effect, most of the time, it's in the true set of dosages where there is an effect. Okay, so if a dosage ends up in our estimator of the dosages where there's an effect, then that dosage is in the true set of dosages where there's an effect 95% of the time. So this is, we're controlling the family-wise error rate at the 5% level. Okay, these two are the same thing. This is just mathese for this. So that's nice, it's good. We're controlling the type 1 error, and it does give us some dosages, and we're kind of happy. There's something that doesn't totally make sense about the set of dosages that we learned, right? So we decided that, that the drug is actually effective at this dosage, and at this dosage, and this dosage, and this dosage, but because of what we did, we decided that the dosage is, if the drug is not effective at this intermediary dosage. It's kind of weird, right? So in other words, if we look at the estimator that we concluded with, we're saying, well, if I give you 5 milliliters of the drug, it's gonna have an effect. 6, no. 7, no. 8, no. But 10, then it'll have an effect again. Okay, that doesn't make sense, right? So there's something nested actually here about these hypotheses, specifically that we really believe that if the drug has some effectiveness at this dosage level, then probably it also has an effect at all of these other dosage levels, higher dosage levels. Okay? And we'd like to take advantage of that for a more powerful test. And ideally, also, we'd like the sets of dosages that we give to make any kind of sense. We're going to give these to a scientist, and they're going to stare at this, and they're going to be like, this is garbage, go back, do your homework better, because what you just gave me doesn't make any sense. So we'd like to take advantage of this structure, and we'd also like to create estimators that make sense. Okay, so this is this nestedness that I was talking about, and we'd like to think about how to use that. Okay, so to use that, we're going to use a, I'm going to go all the way over here. I know, sorry, videographer. And I'm just going to say the way we solve this problem is with a technique called sequential rejection. Okay? And this is a sort of Goman, and Solari, I think maybe 2007. And this is a nice framework for thinking about these kinds of problems that's very general. So the way it applies in this particular situation is we're going to draw the 5th percentile, not the very small 5th divided by m percentile, but the full 5th percentile. We're going to draw that dotted line. And we're going to start from the right side. We're going to look at this observation. Is it below the 5th percentile? Yes. OK, keep going. Yep, it's below. And now we get the first guy that's not below the 5th percentile, and then we stop. That's the algorithm. OK, so that's going to give us all of these guys. So this is sequential rejection. And it turns out that this algorithm, you start from the right, and you wait until you get above the 5th percentile. And the first time you get above, I screwed this up. First time you get above the 5th percentile, that's when you stop. Right before you get to the 5th percentile. So in other words, here I'm still below this dotted line, so I keep going, and now up. This is the first time I got above it, and then I stop. So this technique, this sequential rejection technique, it also controls the family-wise error. And I'd be happy to talk more about how that works and why that works. So here's sort of a hand-wavy explanation. You test this at the 5% level. You don't need to do any multiple hypothesis testing because I've only tested one hypothesis. OK, but now I'm going to do a second hypothesis, this one. OK, and let's say I reject because I'm at the 5% level. And you're like, well, you can't reject at the 5% level. Here it's a second hypothesis. But I say, no, no, no. I'm actually only doing one hypothesis. Because if I can reject this hypothesis, then I can reject this hypothesis for free. So I can pretend I never even did this hypothesis test. That's a little bit of a hand-wavy explanation. But you can get a feeling for why this might work, this kind of sequential starting from the right kind of idea. It does work. OK. But Wes and I were disappointed because we still didn't get the truth. The true set of dosages in this fabricated example where there is some effect is these dosages. And we only got these dosages with the sequential rejection. In fact, there was a dosage that Bonferroni was able to get that we weren't able to get. We were sad. And this is not just in this fabricated examples. We looked at a bunch of simulated data, a bunch of real data, and we found, like, in many cases, you sort of by eye, you could see, like, oh, I think, the change point is here, but the sequential rejection algorithm ends up here. So what's kind of going on here? One way to think about what's going wrong is that the sequential rejection algorithm, it works strictly from the right. It stops at some point. And after it stops, all of the data that's to the left, it just ignores. Right? We'd spent all this money to get all these dosages. We're starting from the right. We hit a single point that's above threshold, and we're like, oh, I guess we're done. Okay? Somehow we're leaving a lot on the table. We got all this information, and we're just wasting it somehow. Another way of thinking about it is the relationship between dosage and survival, we kind of actually think it's smooth. That goes along with this idea of nesting. If we really think that at this dosage it's effective, then it's effective at other dosages. Somehow we're saying that relationship between dosage and survival, there's some flat line, and then something starts to happen. There's some action, there's some smoothness that's happening here. And these observations don't reflect that. Right? They're all over the place. There's a lot of noise. But actually, if we think about what we think the real relationship between dosage and survival is, it's smooth, which means that sort of a better estimate, maybe, of what the effect at this dosage would be, we might be able to get that by averaging in neighboring observations. Right? Anytime you have noisy data that looks like this and you want to actually know the true relationship, I mean, the first thing you kind of think is, well, let's just smooth it. So Wes and I were like, well, let's just smooth it. And so we tried that. So we ran this little smoother on the data, and we ran like an exponentially moving average kind of thing. So basically, we started with this, and then at each step, you instead of taking this value to be the observed value, you take it to be an average of the value you had before and the new value. So maybe you take like, half an average, like say just maybe the literal average, like between what you had before and the new value. There's lots of different kind of variants of this smoothing. So you end up with something that's just a little bit smoother than what you had before. Okay?
Jackson, why do you smooth from the left?
It's a good question, but I'm not going to tell you yet. It's a secret. So in theory, she has a good point, right? I'm saying that I'm doing this exponential moving average from the left. In particular, this value, it's only going to depend on the observations that are to the left of it. Okay? It's not going to depend on observations to the right of that. And that's going to have to do with this nesting. In order to pull this off correctly, it's going to be really important that the way we smooth only looks at the left. And we'll see how that manifests. So the first thing we did is we just ran this arbitrary smoothing process. And then we ran the sequential rejection, except instead of running the sequential rejection technique on the original observations, we just ran it on the smooth observations. And it worked great. We were very happy. And we were able to get the right answer many times. But every once in a while, we would find that the type 1 error was not conserved. Some particular choice of the smoother, for some particular null distribution, something would go wrong, and it wouldn't work, and the type 1 error wouldn't be conserved. It's not so surprising, right? Because the sequential rejection algorithm, it's assuming that the observations we give it, if they're null, if they're really null, they arise from this null distribution. Okay, but after performing the smoothing, that's not true. Even if all of these dosages are actually null, this observation is now an average of a bunch of null distributions. It's not a null distribution itself, so maybe it has a smaller standard deviation. Something has changed a little bit. The smoothing process actually changes the distribution. But because our smoothing algorithm only looks to the left, and because of the monotonicity, we know that if this is a null hypothesis, all of these are null hypotheses, which means that we can actually simulate what the distribution of this smooth value would be under the assumption that all of these are null hypotheses, either by simulation or in some cases analytically. So we can actually correct for the fact that this smoothing does change the distributions a little bit. And we detail how to do this in the paper, but basically you end up moving these, first you smooth and then you have to move them a little up, a little down, kind of, sometimes they move one way or sometimes they move another way depending on sort of, how the smoothing process affects the distributions. But because we totally understand the distribution to the left under the null hypothesis, we can basically understand what this distribution should be if all of these guys are null. And so then we can figure out a corrected p-value. Okay, so the upshot is, first you run some kind of smoothing that smooths from the left, has to smooth from the left. Then, because we only smooth from the left, we can make a correction based on how that smoothing sort of affected the distributions. And then we run sequential rejection from the right. Okay? And you can see in this completely fabricated example that that does actually work in this case. So once we run these smooth values, we get these smooth things, and now we run sequential rejection to the right, and we stay underneath the 5% threshold all the way up until we get into the S-bar territory, the true nulls. So it works in this fabricated example, and it works on real data, sort of in our experience, and also in simulated data. And I'll show some plots at the end that kind of bear this out. Yeah.
What assumptions does the smoothing have to satisfy?
So for the situation of the sequential rejection, the smoothing has to. The only requirement is that you smooth from the left. So this value can only depend on guys from to the left, and that's because under the hood, the sequential rejection algorithm. Basically, it's able to deal with any kind of dependencies in the nulls.
But it can be any kind of function whatsoever, and it should be some kind of averaging.
Yeah, amazingly, for the SR, it could be anything. So in fact, you don't have to do a smoother. You could anti-smooth if you wanted. So what I've said is that we have this belief that it's sort of smooth, and we're going to use that belief to make a better algorithm. But actually, even if that belief is completely wrong, the algorithm is still valid in the sense that it controls the type 1 error. Okay, so what is this smoothness really doing? We're using this prior knowledge not actually to sort of make a better guarantee about type 1 error, but actually just to increase our power. So in practice, what you can show, and well, that's a little strong. We don't have any theoretical results about this. But in practice, what we find is, if it's really true, that the relationship between dosage and survival is smooth, then this test will be more powered, which means we'll be able to find more true discoveries. No matter what happens, we're going to control the type 1 error. But if the smoothness assumption is right, we're going to get less type 2 error, in the language of, that Alex presented earlier, meaning we're going to be able to have fewer misses. Yeah?
So in practice, would you say actually that your null hypothesis would be that there's up to some effects, some like small effect size? Because it seems weird that you have smoothness, but also that like there's no effect up to a certain point.
Yeah, you're not wrong. So when you really want to do this, you have to think about the fact that you're not interested in effects that are small, right? The real truth is there's some relationship between survival and dosage, and what you want to find is not the point where it goes from being no effect to some effect, but you want to find where it goes from being some effect to an effect that's at least as big as something you care about, something clinically significant. So there is really smoothness in the whole thing. We made this sort of fantasy that, to sort of keep things simple, that it's either no effect or some effect. Really, there's an effect at every dosage, and you need to sort of correct for that. But it's sort of more of a technical issue and not one that actually is a problem in practice.
It's an interesting general point that the null hypothesis is often implausible.
Yeah, it's completely absurd, right? That there's no effect here.
In some cases, we might have a prior that a bunch of things are
really, truly not effective.
Many others like yours, it's sort of implausible.
Yeah, I mean, I feel like if I got enough data, I could convince you that there's a difference in the intelligence of children who eat Cheerios and children who eat cornflakes. You know what I mean? Like, there's an effect between everything and everything else. So, and that's something you come up against again and again in kind of frequentist statistics. And so you always have to be careful, to make sure you know what you're actually talking about and thinking about effect sizes and taking that seriously. Otherwise, you're really in danger of kidding yourself. So, but you can do that in this setting. All right. So this is basically this is the heart of the idea. To deal with smoothness, the answer is you're going to smooth and correct. And specifically, you're going to smooth from the left. So big idea, you got some data, you have some kind of nested relationships between these hypotheses. There's a bunch of hypotheses. For each dosage, you have a hypothesis that you're interested in testing, which is that at that dosage, does the drug have an effect? But they're related. So what you do is you take your observations, you smooth them from the left, you perform some simple correction, and then you run a sequential rejection from the right. Ta-da! All right, so that's for section 3. Okay, now, yeah.
Does this procedure lead to a different answer if I measure more dosages? If instead of measuring every 1 milliliter, I measure every 0.1 milliliter?
It depends on how you smooth, right? If you smooth correspondingly, then it's not going to make a big difference. So the point is in some ways right at the heart of why it is important to smooth. So let's say you measured, instead of every milliliter, you measured every nanoliter, okay? So now you're going to have an observation at all these different dosages. If you run a sequential rejection algorithm, it's almost guaranteed because of technical noise that at some point way over here in the high dosages, you're just going to totally by chance get some experiment where you get some extremely high survival rate. It means nothing, right? It's just noise. You have to smooth if you're going to take more dosages. You have to take advantage of this or your power is going to be shot. So the answer is, of course, you have to smooth correctly in order to get the right answer. So if you're going to increase the number of dosages, you better smooth the right amount more. And what is that? I don't know.
Or maybe the right amount less. Smaller window.
If it's smaller, if you have more, you need to make sure that that any noise that's on the line is getting controlled. I'm not sure. It's not clear in my mind.
More aggressive in some sense.
Yeah.
One might imagine that if you used something like an empirical autocorrelation, that there could be some way to
Automatically figure it out.
There's some result that with something like that you get the most powerful smoothing.
Yeah, that's not impossible. That's interesting.
It really depends on the noise.
It definitely depends on the noise, on this null distribution.
Yeah.
How much of a mess is it,
But it feels like it's related to the autocorrelation.
Yeah, for sure. Any other questions about topic 3? All right, so just to get clear here, it is, it's 10:37. And I can go until 11. That's when, okay, great. Fantastic. All right, I'm going to go all the way over here. So the first extension that I want to talk about is sort of a simple extension in some ways. Well, okay, maybe it's not that simple. FDX and FDR. So you remember in Alex's talk, he described that the most stringent thing you could ask for out of a multiple testing hypothesis algorithm is that it controls the type 1 error. Okay? Meaning that most of the time, every single hypothesis that we reject, that was the right thing to do. Okay? That's a very stringent requirement. There's other things we can do. So I'm just going to review those here in kind of the language that we have here. False discovery proportion is greater than 10% is less than 95%. So this is FDX, an example of an FDX. Remember the false discovery proportion, it's the proportion of the discoveries that you, the hypotheses that you rejected, which you should have rejected. Okay, that's what the false is. So FDX controls, if you have an algorithm that controls FDX, it has to satisfy this inequality.
What does the X stand for?
False discovery exceedance. Here's the false discovery proportion. You want to make sure that the chance that it exceeds 10% is less than 95%. So to control FDX in this kind of nested situation turns out to be basically trivial. You smooth, you correct, you run sequential rejection, and then you just take a few more. So let's say you were able to reject 9, then you can actually reject an extra 10. If you're able to reject 90, then I can reject 100. And that works basically just because I feel confident that these ones were right, and I just need 90% of the ones that I have to be right, so I can just add a few more, and okay, that's fine. So FDX is not so hard with this kind of a scenario. The false discovery rate control, that's saying the expected value of the false discovery proportion is less than whatever 5%. So controlling this requires a little bit of a different approach. It starts the same. We're going to smooth from the right. We're going to make a correction. But instead of running a sequential rejection technique, we're going to run a technique called Dagger, which is an algorithm that I don't have time to totally go into the details about from Mike Jordan's lab in Berkeley. And basically, it's an algorithm that deals with these kinds of nested hypotheses. But instead of trying to control the type 1 family-wise error, it's going to control the false discovery rate. And sort of the cool thing is that you could just run Dagger directly on the raw data. But we find that if you first just smooth a little bit and correct, then you actually get much more power. And I'll show some plots for that later. So the big picture is the same, even though the details, instead of using sequential rejection, use this Dagger thing, which is a cool thing. OK, so that's the first extension I want to talk about. This is all section 4, I guess. And then the second thing is I want to say it's not just for chains. So what do I mean by chains? The relationship between these hypotheses here, they fall along this one-dimensional chain. If I can reject this one, I can reject all of these. If I can reject this one, I can reject all of these. It's a one-dimensional space of hypotheses that I'm interested in. A chain of hypotheses, if you will. But you could consider situations where the logical relationships between the hypotheses doesn't fall along a chain. So for an example of that, we're going to turn to problems in differential expression analysis for single-cell data. And I'm going to erase something, and I don't know what. Decisions.
Do you want me to erase while you talk?
What do I want to erase? I don't know.
Do you want help erasing?
I think I'm good. I think I'm going to erase this.
More work.
Thank you, though. I appreciate it. Yeah, I think I'm going to erase this part. Hold it in your mind. It's actually all I've done. But it works great. Bonferroni's cool. This is what type 1 error is. Family-wise error. Okay, it's all locked away in your minds now. Okay, so let's think about differential expression analysis. So let's say we have two different groups of cells, and we're interested in how they are different. Okay, what does that mean? Well, one way we can think about how two groups of cells are different is we can look at their gene expression. So maybe we do some kind of single-cell RNA experiment, which gives us some kind of information about which RNA molecules are present in these two different groups of cells on a cell-by-cell basis. So for each cell, for each gene, I get some indication, some measurement, which tells me something about the extent to which the RNA that corresponds to that gene is present in that cell. Okay? And then I can ask questions like, well, this group, group A over here, is it different in terms of how much it seems to be expressing the gene NPY from this group over here? Do they express it differently? It's a question. Or I could look at some other gene. I could look at VIP. I've been doing stuff from neuroscience, which is why I'm saying neuroscience-y genes. But whatever you're interested in, whatever gene you're interested in, there's this question. Does this group of cells express this particular gene differently from this group of cells? So it's a little bit of a, dare I say, myopic point of view into how different cells are different, to look at one gene at a time. We'd like to be able to think about these relationships, what's going on between these two different cell types at a little bit of a higher level than one gene at a time. Right? We know that there's all these huge, complicated networks, all kinds of stuff going on. So there's different ways to think about that, but I'm just going to give you one simple story. One could use gene ontologies. Okay, so we have a bunch of different genes of interest, and a kind of a cool thing, I guess, that gene ontology does is it draws circles around those genes. And it says, oh, well, these genes, they all have to do with metabolism in the liver. And these genes, they all have something to do with metabolism somewhere. Maybe these ones have to do with metabolism in the kidney. And these ones have to do with bilipid membranes. Okay, so it draws all these different circles around genes and says, this group of genes, they're related in some semantic and interesting way. So at least in theory, this should offer us a way of thinking at a higher level about what the differences are between these cell types. So for example, instead of testing the hypothesis that this particular gene is differentially expressed between group A and group B, we can ask the question that at least one gene in this group is differentially expressed. Now that's nice because even if actually there's a relatively small effect in terms of the difference in each of these individual genes, we may be able to resolve it and say, I don't know which gene it is, but something's going on here. There's an effect, for this group. Meaning that in some level, the way that metabolism is happening is different between these two cells. I don't know what gene's responsible for it. I don't have the statistical power to resolve that, but I can say something about this group. Okay, that's cool. So thinking about these hypotheses instead of at the gene-by-gene level, at the group of genes level. Now, this induces a logical structure on the hypotheses you're testing. Because if I can convince myself that there's at least one gene in this group which is differentially expressed, then certainly it's true that there's at least one gene in this bigger group that's differentially expressed, because this bigger group is a strict superset of the smaller group. Okay? So before we were thinking about dosages, but now we have a similar sort of story with groups. This is another case where sort of this nested idea makes sense. So I'm just going to give these names. I'm going to call this, I guess I'll call it group 1, group 2, call this one group 3, group 4, okay? So if I can reject the hypothesis that these genes are expressed the same, this group of genes is expressed the same between the two cell types. In other words, if I can convince myself that these genes are different, differentially expressed, then I know that there's at least some genes in group 2 that are differentially expressed. Likewise, 3. And then 4 is just kind of hanging out here. It has no relationship to anything, actually, in this case. Maybe we have some other group, 5, which includes all of them or something. Okay. So now what we have, instead of a simple chain, is, and we could have another one, 6, just to make it fun here. 2 needs to be a superset, so whatever. Can be like this one. Okay. we have these arrows that represent inclusion. And if I can reject if I can convince myself that there's differential expression for these genes, then I know there's differential expression for these genes because it's a superset, so on and so forth. So what are we going to do in this scenario? We can take observations for each of these groups. They're probably going to be really highly correlated because we're probably going to have one experiment, but the hypothesis test for any given group of genes is going to use the same data set, and so all these p-values are going to be super correlated, so we need to be able to deal with correlation. But that's okay. We can get an observation for each group, and then we want to test all these different hypotheses. Hypotheses 1, 2, 3, 4, 5, and 6. Knowing that if we can reject this one, we can reject those two. Okay, so it turns out everything that we talked about in this talk so far goes through basically unchanged. So what you can do is you can smooth from the left. What does that mean in this case? Now instead of maybe averaging your observations from 2 just from one guy, you can actually average from all of its ancestors. Okay? You need to make a correction for the fact that you've done the smoothing, but that's no problem. And then you can run a sequential rejection technique from the right. In this case, the sequential rejection technique is a little more subtle because it's no longer along a chain. You just can't go until you find this thing because there's this different stuff, but it's okay. So Major and Goman. Oh boy, this is not good. 2011, maybe? I should know this, but I don't. Showed that for these kinds of graphs like this, there's a simple sequential rejection technique that handles it. Actually, a couple variants, but they all seem to do pretty well. That allow you to run a sequential rejection technique from the right on this kind of graph-structured, nested hypothesis situation. So again, the story is the same. You smooth from the right, you make a correction, and you run a sequential rejection. Or you can also run a dagger from the right. Yeah?
So for this to go through, do you need to assume that the per-gene differential correlation, the differential expression metrics are independent under the null or something?
Yeah, you don't. And that's one of the wonderful things about sequential rejection. Sort of under the hood here, especially in this graph case, there's a little bit of a Bonferroni magic happening. And one of the wonderful things about Bonferroni is that as long as you sort of get things right on the p-value by p-value basis, even if the p-values are super correlated, you're not going to screw up. So, I mean, the short answer is you can do it, even if there's a lot of correlations in your p-values. The long answer is, you need to be careful. I'm happy to talk more in the discussion about exactly how this manifests. But yeah, obviously there's going to be correlations between all these tests because they're all, this test is treating a bunch of genes. This test is treating, a bunch of the genes, some of which are the same gene. So you better hope that even if these, the observations here are correlated, that's not going to hurt you because they're probably done from the same experiment. Does that answer your question? It does. Okay, cool. So yeah, that's a big point. With Dagger, it's a little more subtle. Basically, Dagger uses some similar things to Benjamina and Hochberg, like Alex described, and so you need to either show that there's this positive regression dependency on a subset condition is met, or do this kind of reshaping to deal with any kinds of dependencies. But even in that case, you can sometimes get it to work. But with sequential rejection, it kind of comes for free. So that's a cool thing. But as Alex pointed out, it's a more stringent requirement. So you're going to get fewer total discoveries if you use sequential rejection because you're controlling the total type 1 error instead of just the false discovery rate, which is sort of a more lenient policy that allows you to get more discoveries. Okay, good. So that's the story here. Works. It's great. I think we're ready to show some plots and then be done. Okay, so now let's see if I can figure out how to make this magic work. Hit a button. It's going to plug in. I can take any questions at this time while I'm figuring out. I think our fine videographer is going to do it. Okay, that's not relevant. This is relevant. No questions. Now I'm supposed to hit a button here. Lectern laptop. I did it. what? It's amazing. And it's going to show it twice so that you guys are just amazed, doubly amazed at how awesome it is. Oh, it's not, maybe, oh, and we're going to have the lights down. Okay, so this is just, some simple empirical results. So I'd like to emphasize that Wes and I are statisticians. Zithian also, we're all statisticians. And we submitted this to Biometrica. We have had limited opportunity, actually, to talk about this with real domain experts. I'm going to be here tomorrow. I'm here today, but I'm a little booked. But tomorrow I'm free all day. And we're really interested in thinking about whether these ideas are actually useful. So these results, these are from simulated data. We do also have some results where we look at real data and it looks fine to us. But we're interested in thinking more about whether this is genuinely helpful or whether it's just sort of a novelty. So if you're interested in that question, maybe I guess talk to Alex. Should they contact you if they want to meet?
Yeah, and on the MIA website there is a link to Jackson's website where probably there is information on how they can also contact you.
Contact me directly.
So either way, yeah.
I don't have my web. I might not have my email on my website.
That's
Or failing anything else, you can contact MIA team, which will include our admin, and it'll all go to the right place. So
Awesome.
the MIA team, which is on our website, you can always email that.
Okay.
If you want to meet with the speaker while they're here.
So these are results on simulated data. So we looked at different kinds of dependency structures among the hypotheses. The deep tree case is pretty similar to a chain case. It's not quite a chain, but it's similar to a chain. Wide tree is sort of these relationships between the hypotheses that are kind of like if I can reject this one, then I can reject a whole bunch this way. Bipartite graph. It's like I have a bunch of hypotheses here, and for each one I can reject some set of these guys. Hourglass graph. I got a bunch of hypotheses that I'm starting with, and if I can reject any of these, then I can reject this kind of bottleneck hypothesis, and then that lets me reject all these other hypotheses. Anyway, there's basically different graph structures, and each of the dotted lines corresponds to some slight different variant on an algorithm that we're using. Maybe we're trying to control this family-wise error. Maybe we're trying to control false discovery exceedance. Maybe we're trying to control false discovery rate. And in every case, the black line shows the kind of power you're able to get at a particular error rate if you first smooth and then correct. So all kinds of different algorithms happening on the right to left part, but on the left to right part, we're smoothing and we're doing the correction, or we're just feeding the raw data into these algorithms. And what you can see is that we're able to make more true discoveries if we smooth first. That's the takeaway here. So basically in every case that we looked at, if you first smooth and correct and then feed that into the algorithm, you get the black power lines, which are more powerful, and we were happy with that. So that's basically what this says. Another plot we're thinking about is think about what the error rate is versus the type 1 error versus the nominal type 1 error. So these algorithms are all guaranteed to control some kind of error at a nominal rate, right? We can prove that they control error at some 5% rate. But in many cases they're conservative, which is to say that even though we say that they control at the 5% rate, they actually control at the 2% rate. They do better than they say. And that seems good, but it's actually bad, because if they're controlling at an error rate higher than you want them to, then they're not doing what you asked them to do, which is control at a particular rate that you asked for. And that has consequences, because if you require less type 1 error, then you're going to get more type 2 error. So you really ideally want something that that's not too conservative. If what it says on the tin is you control the error rate at 5%, then you want to control the error rate at 4 or 5%. So these graphs kind of show similarly what kind of things you get, what kind of error rates you get. So on the x-axis is the nominal, what it says on the tin, what kind of error you should expect, and then on the x-axis is the type 1 error rate that you actually get. And sort of bizarrely, what we want here is we want higher error rates. And again, you can kind of see that the black lines have higher error rates. They're able to achieve more error, and we're happy about that. And basically, that's because they smooth the data, and so they're able to find more discoveries, which is good, and that's at the cost of higher error. So those are sort of the two ways of thinking about it that I wanted to present. And that is, I think, everything I wanted to say about nested hypothesis testing. And now I think we open it up for questions, but maybe first we let people go. Is that how this works?
Questions.
Questions first. All right, beautiful. Yeah.